Okay, this is section 2.4 of the AP Biology course and exam description, which in our book is chapter 5, section 1. And we're going to be looking at plasma membranes in this part of the chapter. And I'll be covering all of these things here. And I found this is not in your textbook or anything, but it's a really good look at what the fluid mosaic model of the membrane is. Remember, for the fluid mosaic model, and in your notes, in your reading, if you haven't already, you're going to find out about how they figured that fluid mosaic model out. So I will leave that for you to read about. But the fluid mosaic model is the accepted model of the plasma membrane, which consists of lots of solid pieces, so lots of different pieces. That's the mosaic part, that when they're all together have flow to them. They behave as a fluid. So in other words, they have flow to them. And I'll refer to this little kind of simple animation. But here we have phospholipids and embedded proteins. That's all it's showing us. It's showing us like a glyco protein here. And so an integral membrane protein and a peripheral membrane protein, and the flow of those in the membrane. And that's what kind of it means by fluid mosaic model. Now, what's interesting is that there's a flow into the two leaflets of the bilayer. And when you put phospholipids like this in solution, they're going to automatically form this kind of double bilayer. It's called a micelle to them. And what's interesting is when they tag these with different markers, they found out there was a lot of lateral motion, but there was not a lot of motion between the two leaflets. So for example, if they tag this one, they wouldn't very often, not 100%, but not very often would they find it down here, and vice versa. But you would, if you tag this one, half a second later, it's going to be in that position. So a lot of lateral movement, not so much top to bottom. Okay, so just to kind of look at that again, that's what it means by fluid mosaic model. We have a lot of little solid pieces here that when they're together, they have a structure, a singular kind of almost structure of that membrane to them, and that they are going to behave like a fluid. All right, so I found this diagram, and I'm going to kind of just really quickly go through it. And then down here, there's it's a cutaway of each one. So by and large, the biggest part of, and let me see if I can find some colors I can use here. Box highlighter, text highlighter. Let's see if maybe just drawing would be better, and I can do a different transparency. Okay, that's fine. All right, so the major portion of the membrane is phospholipid. So the phospholipid is going to consist of a phosphate group and glycerol, and sometimes some variations on that. That's why it has the R group there. And so there's various different types of phospholipids that make up the membrane, but the head of it is going to be and I'm going to go through this I'm probably going to do most of the notes on the next pages, but this is important. So the head is going to be hydrophilic, meaning it interacts very well with water. So we'll find lots of water here, H2O, and we'll find lots of water right here, H2O. But if I came outside and it's getting dark pretty quickly, but hopefully but if I try to put H2O here, it'll pop back out. So I can kind of just show that we don't have, I don't like that to large text here, but something like that. H2O is not found in that part. So the tails are hydrophobic. So it was like light and then all of a sudden it just got dark. So these tails are going to be hydrophobic. And altogether, we can say that the membrane itself, or the phospholipids, have this characteristic to them that is called amphiphatic, which means that they have like if you think of amphibian as double life. So it means that they have like two very contrasting parts, where you have the hydrophobic side and the hydrophilic side. That's the amphiphatic nature of the membrane. So part is going to be hydrophilic part and hydrophobic. Just make sure you know that the heads are hydrophilic, so we'll find water close to the heads, and the tails are going to be hydrophobic, so they are going to exclude water. That's why I have that there. Let's see, what else can we see here that I want to point out, and then I'll go through on the actual here we go. Oops, that changed my text. Did not want to do that. All right, I wanted to have my drawing. Cammie's a pain like this. You got to keep selecting different things for your palette. Okay, so here, let me find a few of them so you could see. They're not like every other, but there's not just a couple. It's embedded throughout. These four, I'm highlighting them, these four fused, and there's many, but just showing you a few of them. 4 fused carbon rings, that is the cholesterol. And remember, the cholesterol is going to maintain membrane fluidity. So if we're talking here, we see our membrane is very fluid, but what's not showing you here is the cholesterol. So cholesterol, depending on how much you have in there, can give you different amounts of membrane fluidity. And different organisms that live in different environments, depending on how much it's important that their membranes not freeze or their membranes not heat up too much, they have different types and different amounts of cholesterol. Let me point out a couple other things on this one. Here it's showing us has a molecule that's moving from a high concentration to a low concentration. And that is accomplished by simple diffusion. It's going right through the membrane. Notice that there's not a channel or anything that is helping it. That is going from high to low right through the membrane. There's not too many substances in the cell that do that. There are a couple that do this very readily. One is oxygen, and I'm not going to, I could do my subscripts easy in Cammie, and one is CO2. Those two do this very easily. There's some others as well, but as far as biological importance, those two are going to be the most, I would say, why is it not letting me do that? Most biologically important. Oh, that's still in subscript. That's why it's doing that. So I'll just make a separate one to show that CO2 also comes through the membrane pretty easily. Okay, so I can just, if I wanted to, copy that over. Oops. What? Sorry about this. Why is it not letting me get rid of that? Cammie. All right. Why is it not going away? edit that out. Okay, I was trying to move these over, but that's not being cooperative. Let me see. Is it still allowing me to... Well, I'll see how dark this gets. Maybe it's not that dark. I don't know. I just felt like recording this tonight, and my kids are being loud, so I ran outside, and it got a little dark. So, all right. And you see some various other things here that I'm going to talk about in more detail. This one's sodium potassium pump. If you remember sodium potassium pump, it's going to be this is actually not correct. It should be 3 sodium out. So the direction of these arrows is wrong here. It should be 3 sodium out and 2 potassium in at the expense of 1 molecule of ATP. So that is a sodium potassium pump. So that's going to require energy. All right, now that we see that all together, let me drop down to the ones here. All right, so just to reiterate about the phospholipids, they are the major components of the plasma membrane. They are amphipathic which means that they have a hydrophilic head, which is the phosphate group. It interacts very well with water. Okay, and then they have a hydrophobic tails, which are made of fatty acids. And those fatty acids, hydrophobic tails, they don't interact with water. They don't like water. They're quite depending on the R group on the phosphate part on the head, there's variations. There's many different types of that varieties that make up the membrane. Different species have different amounts. I can't spell varieties. It's showing us the difference between a saturated tail and unsaturated tail. So like if it has that bend to it, that's going to be because it has an unsaturated part. And then if it's straight, because they're saturated. Let's see if there's anything else we need to say about phospholipids. I think that's good. But remember, that's the major component. So there's going to be a practice AP question here that we're going to do that talks about the membrane. All right, next one here is cholesterol. And cholesterol consists of, structurally, is going to consist of 4 fused carbon rings. That tells us that this is a steroid. And things like testosterone and estrogen are going to be involved with, or cholesterol is going to be, those are going to be synthesized from cholesterol. But in general, cholesterol is going to be used in the membrane to maintain the correct, I cannot spell tonight. I always say that. Correct membrane fluidity. Too much, it's too fluid. Not enough, too brittle. So it gives it the right membrane fluidity. Therefore, cholesterol is an essential part of our body. Some people have too high of cholesterol, and it's usually not that related to diet. Sometimes your diet can change your levels of cholesterol a little bit. But there are some people that have genetically predisposed to higher cholesterol amounts. And there are some indications that you find those in things that, like in the plaques that cause heart disease. But there is some controversy if that's the case or not. Just because you find it there, it's essential part of membrane. So it might be involved in kind of in fixing the damage that's happening to vessels and that. But it's, you know, one of the markers of health that they measure when they are taking your blood work. All right. Good one here. Just looking at not necessarily a membrane component, but how something can get through the membrane by simple diffusion. So very few things, very few substances can go directly or simply diffuse through the membrane. That's important to remember. But there are a couple that are very important: oxygen and CO2. To a certain extent, water, but not too much water, can get through. So the reason why things can get right through the membrane is that they either have to be small and they have to be non-polar to get through the membrane. So non-charged. These things are going to get through the membrane easily. So if it's big, if it's polar, if it's got a charge on it, like ions, like sodium and potassium. Water is polar, but it's also pretty small, and only small amounts of water can get through. So I'm going to leave it off and just say that our two most important components that can go right through the membrane are oxygen and CO2. All right, and that brings us to the next one, which is how do large amounts of water get through the membrane? And there's another section of this chapter that I'm going to show this in more detail, but they discovered that a lot of cells have these channels, and since they're pores that bring in water, they just called them aquaporins, which, you know, makes sense. Sometimes stuff in biology is simpler than you might think, and so an aquaporin is literally a water channel, and it is going to bring in water from high to low. It's a very specific to water. We would find a lot of these in our kidney cells, but other cells as well. Again, we're going to talk more about aquaporins in a different part of the chapter, but that's an important part of the cell. And all right, this one here, if we look at it, it's going from high to low, but it needs this transport protein, kind of like it needs a little bit of help. It needs a door to get through. It can't just go right, like pass underneath the door or something. It can't just pass through the wall or the membrane. So the transport protein, in this case, this is something that's involved in something called, you should remember this, facilitated diffusion, which, remember, is still diffusion. It's still going from high to low if you look at that, but it requires the protein. And some things that can be brought into the cell this way, there's quite a few, but I'm going to just talk about a couple. Glucose, for sure. Glucose transporter. Glucose is too big if you don't have this kind of door to bring it through. There are some, like, for example, chlorine or other ion transport. Like what happens to bring in chlorine, which sometimes when it's mutated can cause cystic fibrosis, chlorine transporter. But remember, it's still going high to low, so that's called facilitated diffusion. Usually things are going to be either too big, too charged, or things that are generally polar. Sometimes if they're polar, they're going to be one of these other things too. So, all right. Important thing to note here is still high to low, still facilitated diffusion, no energy required. In contrast to this one here, if it's a sodium potassium pump, and like I said, if this is outside the cell and this is inside the cell, these direction of arrows are wrong. So I want to correct that. I really like this diagram though. There's going to be 3 sodium out. So if this is outside the cell, and this is in, it should be 3 sodium out and 2 potassium in, at the expense of 1 molecule of ATP. So let me write that in here. All right, sodium potassium pump is going to be used to maintain gradients, electrical gradients across membranes, and that's going to be especially important in muscle and nerve cells. We'll get into more details in another section. It's used in a lot of cell types, but muscles and nerves especially, and that's going to be help keep impulses, whether those are muscle or nerves, moving. And so your brain is highly dependent on a sodium potassium pump. So if you don't have a couple different things, if you don't have enough sodium, if you were to not have enough potassium, or not have enough ATP, and like if you don't have glucose, you can't synthesize ATP, your brain will start to shut down. The sodium potassium pump will start to shut down, and the brain will stop functioning. So it can become a very fatal. Problems with sodium and potassium and low energy can become problems very quickly. And they're going to be at the expense of 1 molecule of ATP. It's a pump. So it's going to pump against the gradient. It's going to go from low to high, and it's going to cost 1 ATP for every pump that it does. All right, the next type that we're going to talk about here is the co-transporter. And co-transporter sometimes is glucose co-transported with something else. We'll talk more about specifics, but it just means that 2 things are brought in together. So like there's a sodium glucose transporter, I believe, co-transporter. In this case, it doesn't look like ATP is involved there. It has it going from high to low still. All right, glycoprotein. Glycoprotein means there's a sugar attached to a membrane protein. In this case, this is an integral membrane protein because it's going throughout both sides. You can see that this protein here is going in, it's going through both sides. And attached to it, we have these sugar molecules. They can be much longer than this. But glycoproteins often are involved in cell-to-cell recognition. So, for example, in your immune system. So they're involved in cell-to-cell recognition and communication. Okay, I'll just leave it at that for this purpose. All right, we're kind of going through this. I'm going to stop right here for a second and go inside, just because it's getting really dark and out here, and don't want to get bit by mosquitoes. All right. Okay. The next part of the membrane that we're going to go through is proteins that are involved in cell signaling. So in cell signaling, there is going to be receptors that are usually going to be membrane bound, and the receptors are going to then receive a cellular chemical message that is going to then activate another response within the cell. Sometimes it's almost like a chain reaction that are going to send a signal. So examples of receptors can be like, for example, there is an insulin receptor. Not insulin itself would be the ligand. The ligand is what activates or is going to match